fragments in one single go. Because the primer is one and the same, they are sequenced at one single time. So that is why this particular NGS, next generation sequencing technology, is called as high throughput sequencing. The same thing holds good if you take RNA, isolate RNA, fragment it into pieces, then these fragments attached with the adapter molecules, and these adapter molecules attached to a fragment on either side, right? And the primers that are targeting these adapter molecules, right, can attach to all the fragments that are there in that particular solution and will result in the sequencing of all the fragments. Now, if you look at this particular RNA sequencing technology, we'll talk about this RNA sequencing technology in detail, how much the data needs to be generated and how that files looks like later on. That are already there in the genome. Please note this point. In a microarray experiment, you can only work with explicit. Whereas in an RNA-seq experiment, you can go in for a novel discovery, for a transcript, or for a new gene itself that is being expressed in a particular tissue or a cell. Whereas here, you have a plate on which you have probes as small.
Sir, problem with the voice. Sometimes it is clear.
So continue.
Let me check out. And if you are sequencing the fragment, applying a primer that is complementary to your A1 adapter and sequencing only in one direction, you're going to generate data only in this particular direction. And if your data is generated in only one single direction, it is called as single. And if your data is generated from both the sides by using primers on this side as well as the primer on this side for both the A1 as well as the A2, then you call it as paired end data. This should be very clear in your case, like, because whenever a vendor asks you, like, what kind of data should I generate? The first thing that he will ask whether it is single end data or paired end data. What he means by this is, like, after fragmentation and library preparation, should he go in for sequencing from one side or should he go in from sequencing from both the sides? This is what it means. Means a single fragment is sequenced from both the sides by using paired end. By single end, if you get what single end and paired end is, fine. You have asked the vendor to sequence it either from one side or both the sides. And how does the data, like, now the vendor will come up and say, I've generated the data look like. Now the data, if most of you are familiar with the format called as FASTA. FASTA format is nothing but a greater than sign followed by a FASTA command line, which actually represents the identity of that particular sequence. Then followed by, the only thing that you'll have is concerned is the sequence should not be in continuity with that of the FASTA command line. It should be only after enter sign or a new line character. New line character in computer terms means you press enter and your sequence should be present only from the next line. A sequence can be detected as a FASTA sequence only if you have the data in a new line after the FASTA command line. If you have a sequence, let us say you have a FASTA command line, and if you have a sequence like this, this particular sequence cannot be recognized as a FASTA sequence. Now, how different is the data that is generated from this FASTA sequence, which you already know? The data that is generated from the machine is actually FASTQ sequence. Now, in a FASTQ sequence, the data, every single read, for every single read, you have the @ sign first, followed by the identity of that particular read, right? It gives you the machine ID as well as some identity characteristics, followed by the sequence, whatever it is, A, T, G, C, right? And likewise, right? Followed by a plus sign, followed by the quality of each of these base pairs. Let it be 100, let it be 200. I'll tell you what the quality is. So now what happens, if we can now go back to the previous segment, we have an adapter here, A1, we have an adapter here, A2. Now you are sequencing it. Now what happens is, when you're sequencing it, you have an option to tell the 75 base pairs or 100 base pairs or 200 base pairs, 150 base pairs or 200 base pairs. Is that okay? 100 base pairs. Is that okay? So it means like you are telling the vendor, once you start sequencing, you have to sequence to that particular length which you desire. 25, or this can be either 100 base pairs, this can be either pairs or 300 base pairs. Now, what I mean by a read is, see, this FASTQ file that is generated will have an identity of this particular sequence, particular fragment. So this particular ATGC, whatever you have given, will go to a length of either 75 base pairs or whatever is your desired length which you have asked the vendor to sequence. So the FASTQ file for each single read would contain 4 lines. The first one starting with @sign, which gives the identity of that particular read, followed by the sequence length, whatever you desire to have, followed by a plus sign, and followed by the quality of each of this particular nucleotide. Now, what do you mean by quality here? See, when a reaction is taking place, you have thousands of fragments in the background that are being sequenced in one single go. That's why it's called as high throughput. So the machine actually tells you how good I could read this particular sequence. Let us say adenine. Like how good I could read this particular sequence as adenine by a number. This number mean. This number is nothing but a Fred score. This number is nothing but a Fred score. Talking about Fred score, it is nothing minus 10 log P. Now, if this is minus P gives the probability of going wrong. For example, if you talk about the probability, the Fred score would become 20, 1 in 100. This becomes 20, means this is to the power of minus 2 again, and this becomes minus 2 into minus 10, that becomes 20. So now what happens, when you have a Fred score of 20, it means that you 100. Now when you have the Fred score increases, as this particular value increases, means that you are more confident in saying that, okay, this particular nucleotide is red. So if it is not that a FASTQ file for each single read, @sign, which is @sign, which is a command line, followed by the sequence, whatever length you desired, followed by a plus sign, followed by the quality of each of the nucleotide that is being read by the machine. Now, some of you must have a doubt for sure, if you're following me, that why is there a plus sign here? That you may need to have some more characteristics when the data is generated. But